All right, this is going to be lab number 2. The title of this lab is Mark and Recapture. It's an interesting lab. It's a techniques lab. Mark and Recapture is a technique that's used to estimate populations of organisms. It could be fish, it could be insects, it could be mammals, deer, mice, what have you. So it's really useful in wildlife management, looking at deer herds, fish populations for setting creel limits, hunting limits, that sort of thing. Some idea of the population size for, say, threatened species. How many are there? Is the population still viable? So it's a really powerful tool. It doesn't even have to be animals, though. As we start going through, I'll put an equation up above the video, which is the Lincoln-Peterson index. It's the equation we'll use to estimate population size. And more or less the way it works, you can't sample the entire population. You can't do a census. It's not economically practical, and it's not environmentally doable. You could come in here with dozers, scrape everything up, and count every single mouse, for example. You'd get a census, but then the place is destroyed, and the cost for man hours to do that sort of thing is just way too high. So you have to have some kind of estimate based on samples, and that's what this is. Basically, the reason it's called Mark and Recapture is because what you can do is take a whole population that you don't know. Excuse me, move it, dog. You don't know its size, and you're going to take a subsample of it and use that to estimate how big the original population was. It's called Mark and Recapture because what you do is you sample the population using whatever. It's fish, you might use a net, a seine or something, or a cast net. If it's mammals, we're going to be doing small mammals, so we'll be using these little Sherman live traps, they're called. You collect some of the animals, and then, that's capture, and then you mark them. For fish, you might put a tag on their dorsal fin. Turtles, they sometimes put notches in the shell. We're going to be using some little Monel ear tags. I'll put a bigger picture up here. But these are little individually numbered tags. I don't know if you can see the number there. It doesn't quite focus that good. But we'll be placing these in the ear. So you have a sample of organisms, in our case mammals, that you mark. You let them go. They mix back in with the entire population. And then you take a second sample, and you can look at the ratio in that second sample, of marked to unmarked animals, and that's the same as the original population size compared to the total number of original marked animals. I'll put a couple little demos up here, maybe in the top above the video, as we get going to kind of see how it works. But it does give you a pretty easy way to get an estimate. It's not an exact count. There's a lot of variation, but it'll give you an idea for comparative purposes. It gives you some idea of what the number is likely to be. There are some restrictions on it when you're using animals. You're assuming there's no emigration, that is, no animals leaving your area, no animals coming in. It's probably not true. There's a few other quirks, equal probability of collecting them. That is, an animal that's been trapped is just as likely to be caught as an animal that hasn't been trapped. You'll violate a few of those assumptions, but the index value gives you something useful for comparison. Now, in the lab, I will post sort of the instructions with the equation, the Lincoln-Peterson index. I will post the data sheets that we'll use, and then I'll make a video or two of how we collect the data. That's what I'm starting with now. We're going to be using the Sherman live traps. There's money in the live traps. Sherman live traps. These are little metal live traps. They have one latching door in the back, and they have a spring-loaded door in the front. What you do is we'll go in here. We're going to take a little bit of peanut butter and rolled oats. That's this mess here. Put that on the trap. There's a trigger plate back here. Trigger plate. I'll use a smaller camera for this. That's a little trigger plate. We'll also probably put a little bit of cotton in the traps. This stuff here, cotton. That way if it gets cold, they don't get hypothermia. Some of these animals end up in traps a lot, so we'll put that in there. Then you close that door and it latches. Then the front, there's this hinged door. Pushes down, and if you push it down, it'll catch. And then it's set. If something comes in and steps on the back, boom, we'll trap them. We're going to take, I think in this sample area, this site, we're going to use, I'll probably start with 25 or 50 traps, haven't decided yet. What we're going to do is put a trap every 10 meters. So what we've got here, it's kind of This is mostly pine woods, and it mixes into some hardwoods back there. There's open field and old cropland here, so it's not a bad spot. It's pretty old, probably trees look about 50 or so. We'll take these traps, I'll flag where I'm putting them, and we'll put them down about every 10 meters. Now when I set the traps, you don't want to just set them out randomly. You want to put them where a mouse is likely to go. Like for example, you don't put a trap in the middle of an opening like this, because mice don't like being in the open. Things fly down and eat them. So mice are always up against cover. So we'll typically put them along the bases of trees, something, like this. We'll put them against down logs, piles of brush that are growing up in the forest floor. That's what we'll do. So we'll set about 25 of these. Then me, then I'll come back every day. You're supposed to check these things twice a day. I've usually found in this area once a day is sufficient. We don't have many daytime or diurnal critters. Mostly it's nocturnal. But I'll check them once or twice a day. When we check the traps, I'll pull out the animal. I'll get its weight. I have a little tube scale to get the weight of the animal. That gives you an idea of its age. We'll put on one of the little ear tags. I've got a special pair of pliers. Just put the tag in there and crimp it on her ear. Write down the number, so they'll be individually marked, and we'll let them go. So you check every trap, mark all the animals, come back the next day, check the traps again, look inside them, and you'll have some new animals that you hadn't caught before, unmarked ones, and you'll have some you caught the previous day or the previous days. Typically, I'll carry this out for about probably 5 days, depending on what we catch. 5 days to 7 days is typical for my trapping sites. And you'll get a population estimate here. So I will give you the data. You will have a point value. What is the estimate of the Lincoln Index for mice at this site this year? Then in the folder, you'll also have a page that will list data for my other study site. My normal study site is right behind the school. It's next to the public library. I've been trapping, doing this market recapture there for probably 60, 70 semesters. So I've got years of data. So you'll get the mouse population just behind the ECC campus, kind of an more urban setting, suburban setting. There's houses all around. It's next to the library, EC3. And then you'll have another estimate from this, roughly half-acre site or acre site here, which is much more rural. It's farmland, cropland, cows over there, soybeans over there, Amish next door. And you'll make a comparison. So what you're going to turn in, I'll give you the data, probably give you the raw data for this study site. I'll give you the actual population index from previous semesters. You'll calculate your Lincoln Index, and then you're going to compare the two sites. I'll try and do a quick run through with the camera at the other site so you can see what it looks like. And just kind of try and explain why the population is different or why it's the same. I don't know what it'll be yet. Let me pan around here so you can kind of see what the vegetation looks like. Lots of pine in this spot here, some cedar, a few cherry trees, some smaller dogwoods, hardwoods back there. Out that direction will be some cropland, and there's some open fields out there. Fairly mature trees. Like I said, about 50, 60 years, give or take. So that'll be kind of the intro data. And then I'll put in another video where we're actually checking the traps. So you'll be able to at least see what we catch, how we process it. That'll be the next video. As I'm collecting the data, I'll have to bring a cameraman down, but they'll follow me around and we'll pull out the critters. Hopefully we catch a few. And I'll show you how we ear tag them, how we weigh them, determine their sex, that sort of thing. That'll be the second video. And then the third one will just be a piece of paper, basically with your assignment, maybe a little wrap up. That's it for now. Take care.